And so now let's discuss the broad goals of a typical single-cell RNA sequencing experiment. And so in terms of single-cell RNA-seq, there's a few different motivations for why people choose single-cell based assays over bulk RNA assays. And so the first, as I alluded to earlier in this lecture, one of the major motivations is that biological samples in general are highly heterogeneous. And so because the bulk assays average molecular measurements over all of the cells that contribute RNA to the bulk RNA assay, then you kind of lose this distinction of all the different types of cells in your sample. And so in principle, with single cell based assays, because you're sequencing each cell individually, and that means that you can kind of characterize what proportion of your sample came from different types of cells, and you can characterize the difference or the variation in gene expression that you might see between different cells in your sample. And so this also, what also falls under this category of kind of deconvolving the heterogeneity of a sample is that these single cell based assays are also increasingly useful for example, identifying rare cell types. So for example, if you're looking at tumor samples, basically in the field of cancer, some people think that some types of cancers are largely driven by a really small rare population called cancer stem cells. And so these cancer stem cells used to be impossible to detect in an unbiased way when you're looking at bulk tumors. But with single cell based assays, it becomes much easier to identify a small population of cells present in your samples. And so the second major goal of single cell based assays is really to perform what's called trajectory analysis. And so here again, the idea is that if you're looking at, if you want to observe continuous processes like differentiation, then with bulk samples, unless you do some kind of synchronization, it's basically impossible to really tease out all of the different molecular events that can happen along a continuous process like differentiation. This is in contrast to single cell based assays, where, for example, you can take pluripotent stem cells, you can start the process of, you can initiate differentiation, and then you can basically just try to sequence tens of thousands of cells as they're differentiating. And basically when you then, the idea of trajectory analysis is that when you then kind of visualize the cells that you sequenced, because they all kind of are unsynchronized, in principle, if you sequence enough cells, there should be hopefully a representative set of cells that were in any given state along the differentiation process. And so in some sense, by kind of looking at a visualization of all the cells you sequenced, hopefully they can form some kind of branching pattern, or in what's oftentimes called a trajectory, which then kind of allow you to trace what happens as you move from one end of this trajectory or this differentiation process that you visualized to another. And so again, this is a process called trajectory inference. So one of the interesting aspects of transcription that people have started to study using single cell based assays is the idea of looking at different parts of the transcription mechanics. And so the idea here is that gene expression, even for constitutively expressed genes, might not happen at a constant rate. That is, some people are investigating the mechanics of so-called transcriptional bursting, which is the idea that again, transcription may not occur at a consistent rate. It may basically speed up or speed down at different times. And so these kinds of processes you can start to investigate with single cell assays, because you can essentially look at, for example, even constitutively expressed genes, and you can observe basically cell-to-cell variability in how many transcripts you've detected for a given gene. And in that sense, you can kind of make some rough estimates as to, is transcription occurring at a constant rate, or is it kind of bursting or going up and down very quickly? And so finally, people are also interested in really constructing and inferring gene networks in different types of cells. And so in the networks lecture, I briefly mentioned that one of the challenges of building co-expression networks is that you typically might need hundreds or thousands of samples in order to accurately build a network, and that doing RNA-seq on hundreds or thousands of samples is very expensive, and so you can't really do that often. Single cell based assays have kind of brought network inference back into style, in the sense that because you can now perform single cell assays on hundreds of thousands or millions of cells now, people have thought that, okay, well, maybe now I have enough samples to really accurately infer gene networks, whereas I didn't back in the days of just collecting bulk RNA sequencing data. And this is kind of true and kind of not true at the same time. So as you'll see in a few more slides, it is true that with single cell based assays, you can profile hundreds of thousands or millions of cells. On the other hand, you'll find that on average, the amount of information that you get on any given individual cell is not really comparable to what you would get from a bulk RNA-seq assay, just because there's such limited amount of material or RNA in any given cell. You end up getting less information per cell out of the single cell based assays compared to bulk RNA-seq. And so in the next few slides, we'll talk a little bit more about the different single cell capture technologies that have been developed. And so there's a countless number of single cell RNA-seq based assays that have been developed. Some commercialized, some not, some more or less at a higher throughput with less information per cell, and some that focus more on getting more information out of each cell, but at a lower throughput, so fewer cells per assay. We'll focus a little bit more on the main categorization of assays based on whether they do what's called 3' tagging, or whether they profile full-length transcripts.